Okay, so last lecture of this particular series, and then we're going to move on in the semester. And keep in mind, we don't need to do midterm grades in 300 level classes, but we're about there. Like, you had the first exam. I think next week technically is about halfway through the semester. So I think it's week 8, and it's a 16-week semester. So next week would be kind of our halfway point. So this would be a great time just to kind of do a gut check on where you're at. If you want to come in, office hours, you want to see me, you want to talk about your grades, we can schedule something. You want to look at your exam 1, walk through what you missed. We could do that on Zoom because I can just load it and I can share my screen. So we'll wrap up this portion. I know we're in Unit 2, but then next week we'll start diving into the nervous system diseases. Next week, just as a heads up, you actually have 2 pre-quizzes and 2 post-quizzes next week. So nervous system has 2 of each. I will put that in the announcement, but I know some of you're like, oh, I got the announcement. It's like the same format as last week with just like updated information. That's intentional. That's how I do it. But you will see that there are 2 quizzes next week, pre-quizzes, 2 post-quizzes. So I kind of want to give you all a heads up, especially those that are coming to class. So immune deficiency diseases. There's 2 different main types. So these are not autoimmune disorders. That's what we talked about last lecture. These are immune deficiency diseases where your immune system is compromised. And there's 2 main types. There's primary and there's secondary. Primary meaning it's inherited, so it's a congenital thing. You're typically born with it. Secondary means that you're infected with it. You acquire it or you learn it at a later point in time. And that's considered acquired or secondary. So the primary immune deficiencies first, very busy slide. We're gonna zoom in on different points in this flow chart throughout the lecture today. But what we're talking about are single gene mutations, single gene defects in one location. And we've got 5 types that I want to highlight, only 3 of which we're going to dive into. But we could either have a defect in the production of B cells, or a defect in the production of T cells, or a defect in both, or we have a defect, primary defect, in the ability to make complement. So let me just, because we're not going to get to it, but let me just pause there. What would you hypothesize is the problem if you have a complement primary immunodeficiency? What do you think you have trouble doing as an individual? Inflammation. See, that's I mean, you guys you're actually learning. Like, just like the fact that you can answer that question without me even going over it, and we're not going to talk about it. That's exactly what I want you to be able to do in this class. Synthesize information. Like, I don't know the answer to that, but what do I know? I know what complement does. And so if you don't have it, possibly, what do you hypothesize? I'm not asking you for the answer. I'm just saying, what do you think you should look at, doctor? See, that's what you're going to have to be doing in the future is that kind of critical thinking. Okay? Or you don't make phagocytes. All right, let's brainstorm about that one. What if you don't make phagocytic cells? Could that be a challenge? What could be some of the possible challenges associated with not making phagocytic cells? Can't dispose of the garbage. There's a lot of debris. Maybe you have inflammation, but you can't get rid of the debris. You probably have a lot of scarring of tissue. Okay. All right. We're going to get into the first 3, but just for sake of completeness, as we don't have as much time, but I wanted to bring them up because you might see them, and you'll be able to, I think, understand what would be the challenging, the clinical sequelae associated with that. But what we're going to do is on this chart right here, we're going to dive into first a B cell example. So this B cell example. So where am I? I'm basically highlighting this part right here on this slide. That's what's right here. So this X-linked immunoglobulina is right here, okay? And we're not talking about this one yet, but this X-linked immunoglobulina is also known as Bruton's disorder or Bruton's disease. So it's a rare genetic disorder. It's X-linked, so it's a disorder that is more common in males than it is in females, and these patients do not develop mature B cells. They don't make mature B cells. Discovered in 1952. So the signs and the symptoms of patients that have Bruton's disease or Bruton's disorder is they may have low or no B cells and plasma cells, but they have a very normal T cell response. And so you can see from this flow chart, from the bone marrow is where we are, right? In this graph, we're in the bone marrow, and that's this pink box. This pink box is the bone marrow. So we're in the bone marrow. So we get stem cells out of the bone marrow that actually differentiate into a progenitor B cell and then a pre-B cell, but they don't mature. They don't continue. So that's Bruton's. If they don't make mature B cells, what do you think some of the problems might be? What do you think some of the problems might be?
There's multiple, but my biggest concern is the fact that cells.
Yeah, if it's antibody mediated, yeah. If it's an antibody mediated disease and you're not making memory B cells, because those are very mature B cells, then every time you see a disease, it's the first time. That would be challenging. You'd have to be really careful. Okay. All right. Some additional B cell examples. So that was up here, the X-linked immunoglobulina. But if we go further down the tree, you can see that there are some selective places where you actually might not make IgA with an IgA deficiency. Okay, so you'll make IgG in that case. You'll make IgM, IgE, but not IgA. Now, remember IgA is primarily found in the blood and on body secretions. So this one actually happens to be the most common primary deficiency, is an IgA deficiency. So you'll get a lot of variability in autoimmune diseases. Patients are more susceptible to arthritis, certain types of cancers even. IgA is associated with the mucous membranes. So they have a lot of recurrent sinus infections, respiratory GI tract infections. They're 2 to 3 times more common to be atopic individuals that produce more IgE than normal, probably because they're trying to compensate for the lack of the ability to make IgA. Most common B cell deficiency. All right, let's look at T cells. So we're on the right side of that chart. So let's go back here. So we just went down this pathway. Now we're going to go down this pathway and look at some of these T cells. Here's CD8 and CD4. Okay, so looking at the T cell side, DeGeorge syndrome is really one of the more common types of T cell abnormalities. So in DeGeorge syndrome, you can see it's circulated there or circled there. You actually lack a functional thymus. So if you lack a functional thymus, where did T cells first come from? Where do the progenitor cells? From the bone marrow. Then they move to the thymus to do what? To mature. Or a fancier way of saying that scientifically is what? Central tolerance happens to the thymus. You're absolutely correct. Immunocompetence. Nicely done. That's a fancy $50 word that you're paying a lot of money for tuition-wise, so you should remember that one. Use that one. Use it. Display that word, immunocompetent, with pride. Okay? because you earned it. Yeah, that's exactly right. So they do not become educated. They do not become immunocompetent. That's also the location where you've got central tolerance mechanisms. So with the lack of functional thymus, T cells don't really exist. So these T cells are, compromised. You don't make helper T cells. You go down to the far right, and if you go down the left pathway, you don't make cytotoxic T cells either. Okay, what about a combo? What about a combo? So this one is very, very dangerous. And so there was a film series, a documentary film series of a young boy named David Vedder in the 80s. And this predates all of you, but he was affectionately referred to as Bubble Boy. So he had to live his life basically in an incubator, like in a bubble, because he had severe combined immunodeficiency. He had an ADA deficiency. He had an adenosine deaminase deficiency. So if you look at this chart, we're all the way up at the very beginning of the table. And if you have an ADA deficiency, you don't make pro B cells and you don't make pro T cells. So let me just back up for a second so you can see the whole graph. We're talking about way up here. We went down this pathway. We looked at Bruton's and then we looked at IgA. Then we went down this pathway and we looked at DeGeorge syndrome. I'm all the way up further on the tree, and we're talking about SCID, severe combined immunodeficiency, and that's up at the top. Sorry, that's up at the top here in this particular figure. Does that make sense where we are? So no mature B cells, no mature T cells. Okay, David Vedder was the name of the young boy. Life expectancy is pretty low because they're very vulnerable. And other than that, they're healthy. They just, if they have a foreign virus or a bacterium, it could just kill them. Stuff that you and I just don't even think about. We take it for granted. May feel a little miserable today. Like, I'll wake up tomorrow, I'll feel better. I'll just go to sleep a little early and, I'll feel better tomorrow. Your immune system is ramped up all night long. Not in this young man. Yeah.
Is SCID like the same thing that those mice had that you were?
Yeah. Okay, you made the connection. And there was one other thing that I asked some of you to tell me to remind you to talk about. Benadryl. Yeah. Okay. Thank you. So great catch. The severe combined immunodeficiency we can use as a tool. And so I shared with you a study that we did where we took human cells and we implanted them into mice. And we had to use a skid mouse, is what you call it. This is what it is. It is purposefully created, and it's a mutant, obviously, but it's purposely created where it lacks B and T cells. So then you can do a xenogeneic transfer to study what would happen with human cells, but in a mouse organism. And that was the work that I showed back in Unit 1. So that was a great connection. Okay, the other thing that I asked some of your colleagues to remind me of is when we were back in Tuesday's lecture talking about hypersensitivity 1, and I told you the whole story about Valentine's Day dinner, right? And the Benadryl that we gave our daughter. And so why does Benadryl work? Because in an IgE response of hypersensitivity 1, there's a tremendous amount of release of what molecule to cause all the allergic reactions? Histamine. And Benadryl is a very potent antihistamine. That's why when you have allergy season, you're looking for antihistamines, okay, to lower your ability to create an IgE-mediated response. So that's why Benadryl and antihistamines work so well. And there are non-drowsy types of antihistamines, like the Claritin family, where they've gotten more sophisticated on, they're just more specific. So Benadryl is just more broad-based antihistamine, and like a Claritin is just a very more narrow, very specific type of antihistamine. Okay, that was all primary. These are all single gene defects. Let's talk about secondary immune deficiency. So in secondary immune deficiency, these are results from another disease. That's secondary. Result from another disease. All right. Why is malnutrition on the slide? Well, that is a disease. Like if you have a decrease in zinc in your diet, it depletes B and C T cell production. That's why zinc is in a lot of these cold remedies, because it upregulates your ability to manufacture B and T cells. Stress. You increase glucocorticoid levels, that suppresses the immune system under stressful circumstances. Ever wonder why? Like I always get sick like right before an exam. It happens a lot in college. I've seen it a lot in the last 2 decades. Students always are getting sick right before an exam. Fortunately for us, you take it online. So if you're sick, you're not really infecting anybody else, right? Iatrogenic causes. Iatrogenic causes, these are inadvertent adverse complications that come from like a medical therapy, iatrogenic. So for example, bone marrow suppression. So if you have like cancer and you're getting radiation or chemotherapy, then you may end up suppressing the bone marrow. And where do our immune cells come from? Bone marrow, right? So that's an iatrogenic cause resulting really from cancer. And then infection. Any kind of infection can lower the immune system because you stimulate an inflammatory reaction, and you're using a lot of energy to fight off that infection versus, being on guard and ready to fight an invader that would show up that's not associated with that particular infection. So the last example, clinical example that I want to unpack today is acquired immunodeficiency syndrome, also known as AIDS. So the etiology is human immunodeficiency virus. That's the etiology. The epidemiology is its transmission from population to population. So HIV is what leads to AIDS, and you probably knew that, but some students don't understand that, because acquired immunodeficiency is a secondary immunodeficiency. It's acquired. It comes from something else. You weren't born with it. If you had an infected mother, it's possible you were born with it, but you were infected from the blood transferred, not because of a gene mutation. So that's the difference, okay? So this is acquired or secondary. If you look at the very first slide, we talked about primaries and secondaries, and this is the big example that I want to use on secondaries. HIV likely arose from a simian immunodeficiency virus and began diversifying in the early 1900s, right about 1930s. It was first found to have hopped into humans in 1959 in the Democratic Republic of Congo. And the most common transfer today is heterosexual transfer, and that's still the case. There's other ways for it to be transferred, like blood-to-blood contact, IV drug use, which is essentially blood-to-blood contact, sexual intercourse, heterosexual being the most common, but homosexual intercourse is also a method of transmission, and then maternal fetal transfer. A picture of what it looks like. I think on your slides, some of that text was truncated. So today I went back and got the original picture. I'm not sure what happened. And so this slide might look a little different than yours, but it's on the video because I wanted you to see everything. So we have a very interesting virus. And so let's take a peek at this. This virus is a retrovirus. So a retrovirus means that it's an enveloped virus that replicates using a reverse transcriptase mechanism. So you can see the RNA that's found protected in the middle of this virus. And it's got all these different layers, like a lipid bilayer. It's got a caspid. It's got this, sort of outer shell, if you will, that has a lot of these transmembrane proteins that are sticking out of it, specifically like GP120. It has a very, very high mutation rate, which is one of the challenges associated with it. Now, what's amazing is in my lifetime, so I remember in 1984, it was 1984. We in primary school, we had to do like current events, and I still remember today. You'd cut out newspaper articles, and you'd have to come in, and each week there was like 3 or 4 kids from your class that would present on newspaper articles, and you actually had to present. You had to read the newspaper article and present. And so 1984, I think it was 4th grade. So 4th grade, you're reading like newspapers are like a 7th, 8th grade level. So I thought that was kind of interesting. I thought this was a really neat activity. But this particular presentation stuck out in my mind. It was a research article about AIDS. And it was really kind of taking over in the San Francisco area where there was a big homosexual community. And so that was at a phase in the 80s, homosexual contact was the most common mode of transfer. And that's not the case anymore. But I remember hearing the presentation from one of my classmates about this very dangerous disease. And I remember all of us being like paranoid, like nobody drank from the drinking fountains like for the rest of the semester. Like, I mean, that's dumb. But you're 4th grade. I was like 10 years old. You're like, I don't want to get AIDS from the drinking fountain. Like that's not correct information, but it stuck in my mind. And for me to see the type of research that's available today and the types of therapies in my own lifetime, the analogy would be in your lifetime, you see that COVID potentially completely disappears. And you're telling your next generation, like, I remember when we all thought the world was over. Like I was, in college or late high school when it happened, and it was like this pandemic across the world. The world shut down. The water in Venice got clear. Like New York, it was clean to breathe in LA and New York. That was in your lifetime. So this is really mind-blowing, the kind of AIDS research that has been propagated and the treatments that we have for AIDS and the vaccines and treatments that we have for HIV. Pretty phenomenal. Okay, so retrovirus HIV-1 is more common of the two, and it's typically from patients in the US, Europe, and Central Africa. HIV-2 is more common in West Africa. And some notable mutation rate information is the mutation rate is about 1 for every 10 to the 5th nucleotides that are copied, which is not a lot of nucleotides. So every 10 to the 5th nucleotides that get copied, there's some switch that takes place. So it's been really difficult for the past like 40 years of research for scientists to figure out how to get ahead of this virus. But what I'm here to talk about is less about the treatments and cures, but you can go down that rabbit hole and it's impressive. And it's more about how it progresses. So it targets the immune system. And so here's the virus. Here's the GP41 and GP20. It's enlarged. It's not really swollen like this. It's just enlarged in this picture so that you can kind of see what it's doing. It needs to find the CD4 receptor. So if it's a CD4 receptor, what kind of cell is this at the bottom? It's a helper T cell. So it has an affinity for the CD4 receptor, and it binds, and as it binds, it dumps its caspid, this P24 caspid, that yellow thing, it shoots it into the helper T cell and infects it. Hijacks the DNA using a retrovirus because it has RNA and incorporates into the genome of the helper T cell the virus. And now the helper T cell manufactures that virus. So what's important for CCR5, for GP120 to see the CD4 is there's a receptor known as CCR5 that is required for this connection to take place, for that docking to happen. And this was a breakthrough in the research on therapies for HIV. Because there's a population, maybe some in this room, that do not have the CCR5 receptor. And so they are immune to HIV. Pretty interesting. So if you go to this link, and I'll let you read through it, but I want to show you the link. Oh, wait a minute. That didn't work out so well. There we go. Maybe I got to do this way. I thought I already clicked on it. Oh, here it is. So this is a link from a blog in Nature Education that is describing this genetic mutation known as CCR5 delta 32 that has an HIV resistance. And so this discovery was really instrumental in a lot of our newer information on how to treat HIV. Kind of fascinating, huh? So the infection progression. So how does it work? Well, up top, you can see that we've got a CD4 positive T cell that is infected. We have a dendritic cell that actually might see that antigen and now present it. It makes its way into the lymph tissue. And in the lymph tissue, it gets propagated, infects the lymph tissue or the lymph node in this case. And viremia is a terminology that means now the virus is at levels in the bloodstream where it's measurable by blood titer. Like you can actually measure the viremia, the amount of virus that's found circulating in the blood. So now you make an immune response to it. So you've got anti-HIV antibodies, you have HIV-specific cytotoxic T lymphocytes, and you start this clinical latency where you can see that you've spliced the virus into your own cells. So this little red piece is the retrovirus that's now been incorporated into your helper T cells. And this clinical latency can last for months to years. It could be 10 years that the virus just stays around. And when the virus decides to come out of latency and starts to replicate excessively, you diminish your healthy helper T cell population, and that's when AIDS kicks in. Acquired immunodeficiency is a status where you have fewer helper T cells to be able to fight off diseases, and the viremia has gone through the roof. Okay? Peripheral lymphoid tissues like the spleen. The spleen is a very important organ in this lymphatic tissue because the spleen hosts about a quarter of the body's lymphocytes. And so it's sort of a key target where you get a lot of HIV virus residing is in the spleen. So I think that messed me up when we did there it is. When I went to the website, I think it kind of messed up the link, but we're almost there. The clinical progression. So on the x-axis, you can see time, and from a graph reading standpoint, when you see these hash marks, it means that this is not sequential because this goes weeks here, then there's a break, and these are years. Remember I was saying it could be months or it could be up to 10 years. But you've got an acute phase, you've got the chronic phase, and you have the crisis phase. In yellow are the number of copies of the virus or the viremia. So you can kind of see immediate exposure, you get this spike in the acute phase, and then you move into this clinical latency. But at some point in the clinical latency, the viremia starts to grow, and you can see the primary infection, you lose some of your T helper cells, that's the blue line. But then during clinical latency, they're maintained to some basal level, but at some point when you have this diminished level of helper T cells is when you get into the chronic phase and ultimately into the crisis phase. And what you end up succumbing to, patients who have HIV, they move out of clinical latency and they're in the period of AIDS, acquired immunodeficiency, which is a secondary immunodeficiency. What they succumb to are what we call opportunistic diseases. So your immune system is compromised, and so you get the flu or bronchitis, and it takes you. It's kind of like what was happening during COVID, where people would have COVID and they became very, very weak, and then they got something else, or they had something pre-existing, and COVID just made it worse. So that's kind of what opportunistic diseases means. Is it sounds horrible, but the reason we define it that way is those diseases are winning because of an opportunity where an immune system isn't able to do the job that it normally does. So the cell type that HIV destroys is what cell? The helper T cell, the CD4 positive helper T cell. And if it progresses, HIV moves to what? AIDS, acquired immunodeficiency syndrome, or a secondary immunodeficiency. Questions? Okay. Don't forget post-quiz tonight. I think we're going to get you home before the rain and the clouds open up. So nice timing with nature. I make a suggestion. Get some hot cocoa, pull out your lecture notes, and study while you watch the pitter-patter of the rain. Doesn't that sound wonderful? All right. I'll see y'all. Have a great weekend. I'll see you on Tuesday.